Hello and welcome to the sixth video on molecular biology, which will be the structure of DNA and RNA. Within this video, I'll be talking about, first of all, what the DNA actually is and its differences to the RNA. Next up, I'll be talking about nucleotides as the main building blocks of the DNA and the RNA. Lastly, and finally, I'll come to its structure and say something about its discovery. Hope you enjoy. So first of all, DNA makes up the genetic material of living organisms, and it's also the master code for protein biosynthesis. It's an extremely long molecule built up of monomer subunits, the so-called nucleotides, which I'll talk about in a second. And the RNA is also built up of nucleotides, but it differs from the DNA. You can see it here. It differs in the pentose sugar, where instead of a deoxyribose, you have a ribose, and in its base composition, instead of a thymine, you have a uracil, and the number of strands, the DNA is a double-stranded and forming a double helix, whereas the RNA is single-stranded. And here you can see a nucleotide. Those are the building blocks of the DNA and the RNA, and the nucleic acids are long chains of nucleotides which store and transmit genetic information. Three principal components, as you can see here, are the 5-carbon pentose sugar, forming a pentagon, the phosphate group, forming a circle, and a nitrogenous base, forming a rectangle. And this is the formation of a nucleotide. It's a condensation reaction, and I've talked about condensation reaction in my video on molecules to metabolism, which is the first one from molecular biology, so you may check that one out. And in general, the phosphoric acid and the base are chemically bonded to a sugar molecule, wherein water is given off, so that's why it's a condensation reaction, which you can see here. And that's also the formation of a dinucleotide, basically. So a dinucleotide is when two nucleotides are linked by condensation reactions between the phosphate group of one nucleotide and the sugar of another. This is a so-called phosphodiesel bond, and it allows to form a chain of nucleotides, which then forms the nucleic acid, a macromolecule of the organic molecules. So yes, moving on to the actual structure. One of the most important parts is that it has complementary base pairings, so the nitrogenous bases are specific to each other, and they have hydrogen bonding between the nitrogenous bases, so adenine and thymine form 2 hydrogen bonds, and the guanine and cytosine form 3 hydrogen bonds. And that's always the case. And of course, in the RNA, you would have the adenine and the uracil. They also have anti-parallel DNA strands because of the asymmetrical structure, and the asymmetrical structure is because of the DNA backbone is made up from alternating phosphate and sugar molecules, and it has a direction, which is why it's anti-parallel, and each strand has an opposite direction, which is connected to the structural property of prime ends. So the prime ends means that the ends of the DNA strands are either 5', so a terminal phosphate group of 5, or a 3', which has a terminal hydroxyl group. So if it's 5', the end of this strand will be a phosphate group, or when it's a 3', it will have a hydroxyl group as the end of it. And as I already mentioned, it's double-stranded and forms a double helix, which makes the most sense due to its atomic arrangement for the most stable energy configuration. And the discovery, this one is the X-ray differentiation from Rosalind Franklin and Wilson. And the main credit was given to Watson and Crick, that proposed in 1953 the structural organization, and they demonstrated through Franklin's X-ray differentiation that it's anti-parallel, has double helix and complementary bases, and exposed outer bases. And this was, as I already said, due to Rosalind Franklin's and Wilson's generated X-ray crystallography. And this was it on the DNA structure. Thank you for listening and have a nice day.